Good morning, good evening, everyone. My name is Dr. Urvish Patel, and I'm the Chief Education Officer and Director of Clinical Research Training Program, Research Update Organization. And let's start discussion on the one of the most popular and unexplored topic, roadmap to publication, or in other term, how to publish effectively. So this is the most basic fundamentals of clinical research which we are going to discuss today, in which we are going to discuss basics of clinical research, what is clinical research among so many different type of research, and also how to publish effectively in a shorter period of span. It's a step-by-step process. It's not a rocket science. Usually what we are doing it in a normal practice is to structure wise decide some topic, and we follow those steps one by one, and we begin with the A step, and once we reach to Z, the step will finish and then publication will come out. So we are going to narrow down the whole publication process in a very short manner. First of all, let me tell you what is clinical research and what type of research exists. So the research means collection of any known information in a particular way to create a new knowledge. Either the new knowledge will be in form of original analysis, or in form of surveys or clinical trials or experiments, or in just form of a qualitative research or theorizing the research. Basic research, basic three type of research are basic science research, which we are doing it in a phase 0. That's a clinical trials on animals, or some of the cellular genetics and molecular level of the research in human being. So that's a basic science research, and usually people are doing PhD in that one. The second one is clinical research, which I'm going to discuss today. And the third one is the translational research, or bench to bedside, plus clinical trial application to population based implementation. So for example, the most common knowledge of translational research is a newly launched immunotherapy or any medicine which cannot wait for finishing 4 phase of clinical trial to reach to FDA approval. So in the shorter duration of the span, they apply that same knowledge to some group of people, some group of patients, which may benefit out of that in a most efficient manner with the reduced side effect. So we are having in COVID some of the drug without approval, right away we use it. Remdesivir is one of the example. Some of the chemotherapy is following the same pattern. Followed by that, they go to the standard process to open it for a whole community or whole group of people, population of interest. But in a beginning phase, they used to apply on a small, very particular patient in order to directly reach from bench, which is theory, to bed, which is application. So let's discuss clinical research itself. So first of all, before we jump into the clinical research, most common issue I would like to explain, and then we will jump to the process or step-by-step approach. The plagiarism. The plagiarism is basically presenting the work of someone else, whether it's a word, idea, data, information, evidence, or any principle, and you name it as your own work. And plagiarism is a most common troublesome factor when you are working with the manuscripts or doing clinical research, because directly, indirectly, sometimes you use someone's material, you don't give them credit, or you don't give them acknowledgement, and then you publish it. And sometimes it's not about someone else, sometimes it applies to your own work, self-plagiarism, I name it. So usually what happens that you are doing some type of research again and again and again, and you utilize your concept from your previously published project. So that will create the issue of the plagiarism. So plagiarism, you make sure that you identify those issues, you acknowledge, you give them credit, or you cite them in order to avoid plagiarism. Now, before we begin into the clinical research and what is the benefit of having clinical research. This is one of the complicated part or complicated the circuit I would like to show you here. It's a two part of the process. One part, if you see, this is internal validity and the protocol going towards internal validity. What is internal validity? Whatever we are doing it as a clinical research, that is considered as internal validity. You choose one population, you choose one sample, you have one question to ask. For example, what is the number of the people who died during latest or recent COVID-19 wave? Then in that case, you are going to choose a small number of people in order to identify what was the incident, the recent incident, or what is the prevalence of COVID-19, right? That's a research question. That whatever you are getting the answer in your study, that we call it internal validity. And then after you have the internal validity, you are going to plan in a way that internal validity you are going to apply to external validity, means whatever incidence, prevalence, mechanism you find out, that will apply to external world or external population outside your study or not. That is considered as the external validity. Internal validity you count it using different type of test. We will discuss that. External validity you can apply from internal to external. But our goal is to make sure to get the accurate answer for in external validity by predicting from internal population. And whatever error we are getting it through do that we call it bias, type 1, type 2 errors, right? So we are through the whole research process, you need to make sure that our ideas which we are discussing, the whole idea of our is to work on a specific population. This is the world. Think about world in a bigger term. And this is your study project. So the whole idea is to work on this small population in a way that whatever response you are getting it, that response or that answers can be applied to this population or not. So this is all about clinical research. You do it in your lab, or you do it in your study, you do it in your observational study or clinical trial, and then you apply it to the external world. Here is the stepwise approach to go through each and every step, and that step will begin from having one basic idea. So I'm going through the whole process, but before that I would like to show you something most important in everything in one page, so you are going to have idea. Everything what you are looking into this red arrows, everything is one skill. So research question identification is a skill, hypothesis generation is a skill, so on. Everything here is a skill. So if you learn one by one each skill, then you can reach to a successful submission. Once you submit, the post submission steps are different that we are going to discuss, but this is the whole hypothesis generation stepwise approach. We start with the research question. Research question means identifying ideal question for your studies. There could be plenty of questions. What is the incidence? What is the prevalence? What is the population of interest? What is the intervention of interest we are going to keep it? Or what is going to be the primary objective of the study or secondary objective of the study? Different type of objective of the study we calculate. For example, is there any association between tuberculosis and cancer, or incidence of tuberculosis or prevalence of tuberculosis, incidence of cancer in tuberculosis versus incidence of lung cancer outside non-tuberculosis patients, right? So what is the mechanism? What is pathophysiology? There are plenty of questions. Questions could be qualitative, which means it won't have a number attached with it. Mechanism, pharmaceutics, pharmacodynamics or kinetics, or maybe quantitative, that means it's having number. incidence, prevalence, odds ratio, relative risk, hazard ratio. So all research question needs to be identified in advance. Then hypothesis generation, then study design. We find out what type of study we would like to plan. Then we are having cohort identification, population identification, intervention identification, comparison identification, and outcome identification. Definition for tuberculosis for internal medicine physician is different than radiologist, then a researcher who are going to use survey to identify, for pathologist who identify that on the basis of lab results, for anatomist who identify them using the autopsies. So the definition of population, intervention, comparison, and outcome is most important. Once you decide your outcomes, primary, secondary outcome, then we jump to introduction part. We jump to find out some particular article describing the previously published article on that, what are the gaps in those articles and what you really want to do. Then we decide the methodology. Then we write research proposal. After we write research proposal, we submit it to institutional review board committee or ethics committee, or we do data usage agreement with some of the committees where you don't require to get the ethical approval. Once everything is done, then data collection start. Now the issue is data collection is a very complicated process. It takes really long time to finish, especially in many of the developing world where data is not collected using electronic health record or electronic data. Then you need to manually collect the data for your studies, especially if you are in India and you are doing ICMR, Indian Council of Medical Research related projects, then you may face this issue of collecting of the data by yourself. So your phase 1 of clinical research finish when data collection finish for the study. So this is your whole phase 1 steps. In research update organization, we make sure that we organize the full data. We do data usage agreement with everyone, and we make sure that until that phase 1, the data collection, we will teach you side by side when you are learning the concept of research as less as within 1 month. Usually when I remember my ICMR experience, it took me 2 years to just finish this phase 1 to collect the data from here to here. Then phase 2 of the research start, and phase 2 research start with the analysis. Once you do the analytical planning, then you prepare ghost tables in which you plan that what are the number, what are the tests you want to do. It will go each step one by one. After that, you interpret the analysis, do the analysis and interpret the analysis using different softwares, SPSS, SAS, R in India, EpiInfo, and so many more software. And then finally you fill up that table and results, table and figures in terms of primary result. So that phase considered as phase 2. That phase is considered phase 2. And that can be, if data is there with you, then that phase can be finished as early as within 1 week. Why? Because then you are having full 1 week to analyze using fancy softwares to write those results. Everything is on a desktop. Nothing required to go to hospital and collect any other information. So that is going to be much more easy for you to deal with the phase 2 from your home only. Now after phase 2 finish, then we are having path A and path B. Path A means you are going to present your work. I'm just writing here. You're going to present your work in either conference whether it's a national conference, local conference, international conference, you present your work in terms of abstract, which is 300 word of a document. You go there, you publish that the day, or I can say they publish it, the conference association, let's say American Heart Association. Once you present the work in American Heart Association conference, they publish it in their affiliated journals. Let's say Circulation, let's say Stroke. So they publish it, and that finish your one phase of your presentation part. But now from submission to acceptance will take approximately 3 to 4 months for conference, because probably, let's say 5,000 submissions arrive all over the world. Probably 3 judges will review that and will narrow down whatever capacity they are having it for people to come there and present. Let's say out of 5,000, 10% or 20% get accepted. So 500 to 1,000 people will be invited to American Heart Association, and many more associations are there. Total, there are 17 to 18 different associations in different field of medicine. Medicine means medicine, surgery, internal medicine, surgery, family medicine, cardiology, pulmonary, GI, hemonc. Total 16 to 17 different subspecialty and specialties are there, in which they have at least 3 to 4 association in each specialties. So from 500 to 1,000 people are invited to present the work, and after presentation finish, then you are going to write the full length of manuscript, which will be your chapter of 20 page to 30 page. And of course, there are many type of full length. Sometimes the chapters are of only 1,200 to 1,300 words to 3,000 to 6,000 words. So you write the full length manuscript and you go to the path B, that you are going to go through the writing of the manuscript and submit it to the journal. Now, when we discuss that path A and path B, this is what Research Update is doing it, that we submit it for the conference first, followed by that we submit it to the full length manuscript. That is what we are doing it, but it's not necessarily that you can do that, because the time duration is 6 months from writing the completion of the project and submission and acceptance of the path A, which is conference. So you can directly write full length manuscript, and then you can directly submit your full length manuscript as well. So you can do that if you want to do that. So presentation and publication. Presentation means conference submission, and conference presentation, and publication means submitting directly to the journal and then publish full length chapter. Now, if you decide path B, then you need to complete the result section, you need to complete discussion writing, you need to complete conclusion, you have to complete take home message and features of the study, or basically what that study is all about. You need to proofread that document multiple times. You have to cite those manuscripts. After citation, you prepare pre-submission document. Now, what is pre-submission document? That means your cover letter, that means your title page, that means your supplementary material, that means your figure in whatever way they are asking, format according to journal, and then choose a journal and submit it. So this way we can finish the full submission, and the full submission means your phase 3. That means now you not only presented your work in the conference, and you can also publish the manuscript. And if we don't want to go like that way, then as I was showing you before, that you can consider as phase 3, because in UK you are getting special points, I think 7 to 8 points if you are presenting in